you for having me. Happy to give a talk today. So as was stated, my interests lie in neuroinflammation and degenerative diseases, and that's what the talk's going to be on today. So this is a project looking at neuroinflammation in the context of a disease, chronic traumatic encephalopathy, which if you're a little familiar with the center, is a really big focus. And a lot of work we do is really centered around that. So today I'm going to give just a general introduction of CTE. I know a lot of people understand what it is being from Boston University, but I like to make sure everybody understands some of the history of it and what CTE actually is, just to get everybody on the same page. Then I'm going to talk about some of my work in neuroinflammation in the context of CTE. We're going to ask the question, is inflammation involved in the neuropathology? And then if so, what neuroinflammatory factors are involved? Because that's a very important question when we're thinking of therapies and biomarkers and understanding the disease in a more thorough context. And then I'll end with some future direction. So first, just going over some of the history and introduction to the disease, chronic traumatic encephalopathy, or I'll just call it CTE for short. Now, we know CTE has been pretty commonplace in the discourse of sports and athletes and head trauma recently, but CTE is not a new disease. It's a disease that's been around for almost 100 years now. This is work by Dr. Harrison Martland, a neuropathologist or pathologist from New Jersey, who first wrote about case studies in boxers, where these boxers of the slugging type were found to have unsteadiness of gait, mental confusion, slowing of muscular movements, and tremors, and he thought this was caused by multiple concussive hemorrhages. So this is even 100 years ago. We knew that getting hit in the head was not good for you. So this has been around for a long time. We are not the ones who invented this or discovered this. The name changed over time, though. So originally it was called punch drunk, and that's still a term that you can find used today, but it became dementia pugilistica, traumatic progressive encephalopathy, and ultimately chronic traumatic encephalopathy to be more of a scientific name for it instead of just something called punch drunk. Now, a lot of this early work throughout the early decades was focused on the clinical identity of the disease. Individuals had exposure to trauma, and then you could say that they had these clinical phenotypes that could be measured by a clinician or a doctor. But it wasn't until the 70s where you had the first neuropathological examination of these cases. So this was the first time by Dr. Griselius and his team seen here, where they took a case study of 15 retired boxers who had passed away and examined their brain. And they could find that the brains actually had significant neuropathologic features. So this was the first time you can say these individuals who exhibited these clinical abnormalities with the exposure to head trauma had something pathologically going on in their brains. That was a really important step in the field. But up until this point, and even past this in the subsequent decades, CTE isn't something or wasn't something that was really viewed as a concern. It was something that, maybe boxers who boxed for way too long would get, or sometimes domestic violence victims were subject to it. It really wasn't a common thing in the public consciousness. But that really all changed in the early 2000s with the case of Mike Webster. Mike was an American football player, played in the NFL, passed away at the age of 52. He played for 17 seasons, so he played a significant amount of time in football. He had very significant impairments, cognitive loss, behavior problems, motor symptoms. And when he passed away, his brain was examined by Dr. Bennett Omalu, who found that he had CTE. Now, this was very significant in the fact that you don't really think someone who plays, American football would get a disease from it. This is a pastime. This is a fun sport. Kids play this. And now it's becoming the fact that, well, maybe my kid can get a degenerative disease by playing this pastime sport. And it really, sort of thrust public consciousness into better understanding this pathology and really understanding what it is in the context of disease, because it really wasn't understood that well yet. And that's sort of where our group comes in, the BU CTE Center. It was formed in 2007 with the idea of better understanding the neuropathologic features of CTE. Dr. McKee and our colleagues came together and wanted to amass a very large case set to study this disease, because up until this point, it really wasn't examined well. The largest study was those 15 brains by Dr. Griselius in the 70s. And to better study neurodegenerative diseases, you need a large sample size. 5, 10, 15 cases doesn't cut it. So the only way to understand this disease, to get an inkling of what it is to amass a very large sample size. And that's what Dr. McKee and her colleague did. And that came to fruition in 2013, where they published this seminal paper in the field, where they looked at 85 different cases of individuals who have passed away and donated their brain, and they were found to have CTE. And they could establish some very important features that we use today, and it lets us understand what the disease is. So it's important to say, even now, you can't diagnose CTE in someone alive. A diagnosis of CTE can only happen when someone passes away and their brain is donated and a neuropathologist looks at it. And what you find in someone who has CTE is called the pathognomonic lesion. It's the lesion or the feature that defines the disease. And that is the perivascular accumulation of hyperphosphorylated tau at the depth of the cortical sulcus. So all that means is they have the protein tau, which we all have in our brains right now, but somehow it gets hyperphosphorylated and becomes sticky and aggregates together. That tau is found around blood vessels in the brain, so perivascular accumulation. And those accumulations are typically found first at the depth of the cortical sulcus, mainly the frontal cortex, but it can start from the other cortical regions first, and from there it'll spread out. And that is CTE. If someone asks you what CTE is, it is the perivascular accumulation of tau at the depth of the cortical sulcus. And that was defined by that seminal first paper. CTE and the lesions are patchy. So unlike other diseases like Alzheimer's, where you can have this very confluent distribution through the laminal cortical layers, you have patchy distribution in certain areas, particularly in the sulcus early in disease. And from there, it will spread. CTE was originally defined as 4 different stages, stages 1, 2, 3, and 4, where stage 1, you have very focal minor pathology in the frontal cortex. Stage 2, you'll start to have a couple more foci of tau in frontal and parietal, maybe temporal cortex. Stage 3, you start to have medial temporal involvement. Their hippocampus and amygdala will start to get involved. And stage 4, you pretty much have brain-wide involvement. Now, as this field evolved from that first 2013 paper, many experts in the field came together in 2015 to have the first consensus conference for CTE. So even up until this point, it wasn't recognized as its own unique disease. Individuals will claim it's atypical Alzheimer's disease, or just normal aging. And in 2015, they convened together with experts across the world of all different disciplines, of all different feelings on if CTE was real or not, came together and did a case series, and they could identify that CTE was in fact unique pathology. The perivascular lesion is unique to CTE and identifies that you don't see it in anything else. So that was 2015. So that's not that many years ago. So that shows you where we are in the understanding of this disease. And that work was more clarified in only a couple of years ago in 2021, where the same people came back together and they confirmed and improved their diagnoses and their understanding of the pathology. So we're still in the early days of really understanding this field. And that's where a lot of the work I'm going to talk to you about today is really understanding the first possible elements that are contributing to this pathology. Oh, and then just one other thing to point out, some of this later work, they clarified that stages 1 and 2 can be classified as low stage and stage 3 and 4 and high. So I'm going to say it both ways sometimes. I just don't want to get everyone confused. There's low stage and high stage disease. So the main question that comes out of this is what causes CTE? That's what everybody wants to know all the time. What causes CTE? And unlike any other degenerative disease, neurodegenerative disease, we know the cause. We know the cause of CTE, and it's a history of repetitive head trauma. Individuals who have this long history of repetitive head trauma have CTE. You won't find any cases of someone who doesn't have this history that doesn't have it. So it makes it a very, very unique pathology. We know what causes it. And this was very, very well clarified and shown by Dr. Jesse Mez, who a couple years ago published this paper looking at the duration of American football players and CTE, where they were able to show that using 266 American football players, many had CTE and they had several that didn't as control cases, that when you're examining the statistics of the likelihood of having the disease, playing a year of football, every year you play increases your risk 30%. Your risk doubles every 2.5 years, and the risk of severe disease doubles every 5.5 years. And they were able to show that playing less than 4.5 years, you have less than a 10-fold risk, and playing more than 14.5 years, you have a 10-fold more risk of the disease. So this really hammers into the picture that head trauma, repetitive head trauma, individuals who receive hundreds and thousands of hits over their career are linked to this disease. So I think that's been pretty well established. We know the cause of the disease. We know head trauma relates to CTE. What we don't know is this middle step. This is what we're working on now. What is this middle step? How do you go from having this history of head trauma to this degenerative disease? And that's where we think neuroinflammation is involved. So that's where a lot of my work is centered on, is understanding neuroinflammation in the context of head trauma and CTE. So first, a little bit about neuroinflammation. Neuroinflammation gets a bad rap. I think it's a very, very important biological element, and it's not always a bad thing. You need it. It's really important. It's a fundamental immune response designed to protect the body. It kills the infection, promotes recovery in tissue, and it's without neuroinflammation, none of us would be here right now. Chronic neuroinflammation that occurs over time, long periods of time, doesn't get downregulated, that's a bad thing. Now, we know that head trauma causes inflammation. How does that occur? So work by Dr. Lee Goldstein and several others here at BU have shown very eloquently that there are acceleration and deacceleration forces that occur when you get hit. Now, I'm talking mostly about American football here, but it can be applied to a lot of other sports. So that quick motion back and forth if you get hit with a tackle is causing the brain to twist and contort. It's under tremendous forces, linear rotational, over a short period of time that causes stretching in the brain and comes back together. That's pretty bad for the brain. The brain is going to be subject to a lot of different injuries, axonal injury, blood-brain barrier leakage, but it's important to say that the frequency of these injuries is really important. Our brains are very, very good at repairing themselves after minor injuries. But many of the sports that we talk about, football, hockey, soccer, boxing, these injuries aren't rare. They're frequent. Hundreds of hits a week, thousands a season, tens of thousands over the course of someone's career. It adds up. And the question is from these injuries, is neuroinflammation involved? Do we think it's involved in CTE? Now, there's been many mouse models that suggest that it is. But it's really important to distinguish, human versus mouse and the severity of these models. Up until a couple of years ago, we had no idea inflammation was involved in the human brain with people with this on the road to CTE. So that was the first study that we did. We needed a definitive study demonstrating inflammation is present in human brains with CTE to even get on the road here to understanding inflammation as a concept in this disease cascade. And the questions we asked in our initial studies were, does inflammation, if it exists, correlate with CTE severity? Does it correlate with exposure to head trauma? And what cells might be involved in this response? And can we relate it to the clinical symptoms? So we set about grabbing a couple of different sample groups here, using samples from the BU CTE Center. It's really important to have a proper sample selection here because we're working with human postmortem cases and there's a lot of variation in human disease. So we picked four different groups: individuals who are control, so they don't have any exposure to contact sports and they don't have any degenerative disease as diagnosed by a neuropathologist. Individuals with RHI without CTE, so RHI stands for repetitive head injury. And these are individuals who played a contact sport, in this case, American football, but they weren't diagnosed with any degenerative disease. Then we had stage 1 and 2, so CTE mild or CTE low, and stage 3 and 4 or CTE high or CTE severe. These were our four groups where we wanted to investigate, is inflammation occurring with head trauma and with CTE? So how do we do this? We have a fully automated digital slide scanning setup in the BU CTE Center that allows us to scan in glass cover slip slides at 400 at a time. So it's a very powerful system to look at histology. And we use this to do fully automated histologic analysis. So slides are stained with several markers and they're analyzed this way on our slide scanner. We focused on the area of the brain called the frontal cortex, the dorsolateral frontal cortex, which is an area, if you remember from before, is where CTE is one of the regions where it first starts. The rationale behind this was it might be more sensitive. If we look in the region where it starts, we might be more sensitive to find subtle changes. Then we use these digitally scanned slides and a program called ImageScope to highlight regions of interest and quantify the amount of pathology in those regions. And we quantified 4 different things. We did a stain for microglia, which are the brain's resident immune cell. There's a sentinel immune cell in the brain. That's IBA1 is the marker for that. We look for reactive microglia. So when microglia become reactive or activated, they will upregulate several markers. CD68 is just a very general marker of an inflammatory response. It's not very specific for things, but it can show you that there is potentially an immune response occurring. We look for astrocytes, so another very important brain cell type with GFAP. And we use p-tau as a marker of AT8. So we're looking at hyperphosphorylated tau, and if I say AT8, that's just going to be the same thing there. So quantifying them across our 4 different groups, we could see the following. So we saw that there was overall no change in the number of microglia across all 4 groups. Again, astrocytes didn't change as well, so the number stayed pretty consistent. What we did see is there was an increase in CD68. So this is our general marker of an inflammatory response or an immune response. And we see that was elevated across disease. And especially it was elevated in people with RHI with no CTE. So these individuals don't have disease, but they have exposure to trauma, and they have more of these immune response cells. So this is showing that trauma itself can cause an elevated immune response in the brain. And we see that potentially scales with disease severity. We can look at this based on morphologic examination as well. Microglia are a very particular cell type that when they become reactive, they can change their shape from a ramified cell. So they have these processes that are very long and filamentous. And when they become what's called amoeboid, they get a much smaller cell body and shorter processes. And we can see that transition of microglia from individuals who are controlled to RHI and then with CTE. Furthermore, we see that microglia seen here in IBA1 can interact with tau positive cells. So this is a neuron that contains tau. We call it a neurofibrillary tangle or an NFT. And then you can see in the co-stain here, microglia are reaching out with these processes and contacting them. So the actual interaction of these microglia with the tau positive cells. This was one of the first reports showing that inflammation was found in individuals with diagnosed CTE. We wanted to actually compare the effects of multiple variables in the system because we're working in human pathology here, and there's a lot of variables to consider. We wanted to understand if exposure to head trauma, the degree of neuroinflammation, the amount of tau pathology, and age of death, how they all function together and account for each other. Because something really important to point out is that individuals who are older have more neuroinflammation as well. So there's a lot of variables here we have to adjust for. And so what we chose to do in help with our biostatistics department is run a simultaneous equation regression. This is a computational statistical model that can test for both direct and indirect effect on predictors on outcome variables. So what does this mean? We have predictor variables and outcome variables. We have 2 variables, exposure to trauma and age that we think are driving, and then we have our outcome variables, inflammation and tau. And what we were able to show is that whenever there's an arrow, it means there's a significant effect on that population. So we were able to see that exposure to trauma has an effect on neuroinflammation that has another effect on tau pathology. So this is showing that there might be a cascade occurring here. We do see an effect of exposure on tau pathology that's not captured by neuroinflammation, but we're only looking at one area of the brain, and we're not accounting for all the data in this study. So there's likely other factors here besides RHI, and we're very aware of that. Importantly, we saw that this effect is independent of age. Age also causes some tau pathology. It's just a normal aging effect. So we know that that's occurring. But the effect that we're seeing of inflammation is independent of age. It's stronger, that effect. Now, what's really interesting, we found that there's this bidirectional effect. We saw that inflammation has an effect on tau pathology, but tau pathology had an effect on inflammation. And it leads to a potential vicious cycle that's sometimes seen in Alzheimer's, where amyloid begets inflammation and inflammation begets more amyloid. So we have an idea, and our existing model is that such. You have neuroinflammation occurring as which happens after head trauma, which causes hyperphosphorylation of tau. Tau itself is inflammatory. When you have more tau in the brain, you have more inflammation. So you start generating this vicious feedback loop where neuroinflammation causes tau, tau causes neuroinflammation, and it occurs over a long period of time. Now, the last thing we looked at in the study is the correlation to any sort of clinical variable. And what we were able to see is that as you have more inflammation in tau, your probability of dementia increases as well. So we can add that to our table, and we can see that there is this effect of exposure causing inflammation, which leads to tau pathology and long-term dementia. So overall, what we're going to show from this original study is that increased reactive microglia do correlate with CTE pathology. They're also elevated in people with just trauma without disease yet. So it's part of the early cascade of the disease. It's not caused after the fact. It might be part of the inciting incident of the disease. The effect of RHI and CT pathology is mediated by neuroinflammation in part. There's other factors as well. We recognize that. Neuroinflammation in tau might exist in a feedback loop, and it's involved in potential clinical outcomes. So this again is in tissue that has been individuals that passed away and donated it. What came out not recently, several years ago at this point, was a work by Jennifer Coughlin looked at PET scan individuals who played in the NFL, and they used the marker TSPO. TSPO is a marker that when there is more reactivity of glia, microglia or astrocytes in the brain, they will take up more of this PET ligand. And then more of this PET ligand is a surrogate marker of inflammation. And they were able to show in NFL players who were alive, compared to control individuals, they had more of this TSPO uptake. So we can see that inflammation is elevated in people who have exposure to contact sports, who are alive. Now, we don't know if these individuals have CTE. All we know is that they have more inflammation in their brain. So you have to be very careful on those assumptions that just because someone has inflammation, they play in the NFL, they have CTE. That's not the case. We don't know yet because there's no way to diagnose it while they're alive. So the only way someone could have a diagnosis of CTE right now is post-mortem. So that's just a caveat. Now, we know neuroinflammation is elevated in CTE. Can we determine if it's specific to the actual pathognomonic lesion was the next question. The reason we wanted to ask this is because if it's not specific to the lesion, is the inflammation that we're seeing occur a brain-wide response to head trauma, or is it specific to the lesion itself? Those are two very important questions here. Is it a brain-wide response, or is there something about the lesion that's specific to the inflammation we're occurring? And this is, again, very similar to the Alzheimer's field, where you have in green here your amyloid beta plaques, and your red, your microglia. And it's very typical in Alzheimer's to see microglia associate with these A beta plaques, whether that's protective or detrimental. That's another talk and another big field of debate that we can have. But it's a pretty important part of the disease that the pathologic features drive a lot of the glial responses that you see. And we want to know if the same thing is happening in CTE. Do microglia associate more closely with these lesions, these aggregates of tau? So our next project was to connect microglia to the CTE lesion. So how do we do that? Well, we use the same digital images from before, but this time we looked within each image and compared them to themselves, essentially. We said, okay, we're going to select lesion vessels, so lesion blood vessels, lesion pathognomonic lesions, and compare them to non-lesion vessels. So we call them control vessels within the same individual. The idea is that if you look within the same individual, those individuals with tau, those blood vessels with tau around them should have more microglia around them. Therefore, you can say that potentially there is an effect of the lesion on glia as opposed to just a brain-wide effect. And when you count the amount of glia around control or lesion vessels, you can actually see that. So you see that in low and high CTE, when you look around the lesion compared to control vessel, you typically have more microglia around a lesion vessel. So you can say that there is a response of these vessels to either recruiting or causing more microglia to come around them. Whether that's before or after tau forms, we're still not sure yet, but it's a part of the cascade now. Another important thing when we're talking about blood vessels and the brain is microglia and macrophages. I keep saying microglia, but it's really important to point out that macrophages, peripheral macrophages, are different than microglia. Although they are very related, microglia come from a different developmental lineage of immune cells. They come from the yolk sac at an earlier developmental time point than peripheral macrophages that come from the bone marrow. So although they are related, they have different functions. But they have a lot of very similar antibodies. So it's very easy to mistake microglia for macrophages. So we wanted to determine, although we see these cells around the lesion, are we looking at microglia? Are we looking at macrophages? And there's a couple different markers that we could use, but we chose to use TMEM119 and look for IBA1 positive, TMEM119 positive cells, and look to see if there's anything IBA1 positive, TMEM119 negative, which would mean it's a macrophage coming in. And we can see that in control vessels, again, in people with CTE, that you get a very good overlap of IBA1, TMEM119 positivity. But when you look at a lesion vessel, you can see that there are cells around that lesion vessel that are IBA1 positive, TMEM negative. And when you do a quantitation of that, you can see that in both low and high CTE, you're getting an influx of macrophages around the lesion. But the majority cell type there is microglia. So we're pretty confident that the majority of cells that we're seeing react here are in fact microglia. However, there is some sort of macrophage component, which is important to understand, and we're working on understanding that more in the future too. And that might be a leakiness of the vessel. It might be another separate peripheral immune response being activated there. So it's a really interesting story that we're still following up on. So just like Alzheimer's and amyloid beta, we can see that CTE also has this reactive glial component specific to the lesion. Now, we're talking about microglia, macrophages being recruited to certain areas. The question, is really obvious. What's causing them to be recruited? What's causing them to move to certain areas? Now we're starting to get into these parts of the cascade here. What are these middle steps, right? What is causing these cells to lead to these neuroinflammatory sites? What is causing them to induce tau pathology? And the first thing we wanted to examine are chemokines. So chemokines are a class of proteins that is pretty indicative for all cell type and all biology in terms of cell biology. They act as a migration signal per cell. They're not specific to microglia, obviously. They're for many different cells, and they act over a concentration gradient. So something produces it, and the gradient goes across tissue, and cells know to follow that gradient towards the thing that's producing it. And when we're thinking about chemokines involved in microglia migration, and glia migration in general, the most prominent is the chemokine CCL2 or MCP1. We'll call it CCL2 because that's a common name for it. It's the most potent glial chemokine. It's a very potent macrophage chemokine as well. It's produced by many cells to bring microglia and other cells to the area where they need to be. And it's elevated in many diseases. So it's elevated in AD, MS, stroke, TBI, and several mouse studies have shown that when you knock out CCL2, it's beneficial. So you don't have a recruitment response of these immune cells, and you have a less inflammatory response and better outcome. So is this true in CTE? And if it is, maybe CCL2 is a therapeutic target that we can maybe harness. So the experimental design we're going to be looking at here is taking three different groups here, RHI without CTE, individuals low CTE and high CTE. We're going to be using tissue from the frontal cortex, again, essentially doing an ELISA to look at just the amount of CCL2 that's in this tissue and compare it to a couple different things. We're going to compare it to the amount of years they play contact sports, compare it to the amount of IBA1 positive cells that are there, and the amount of CD68 positive cells that are there. And what we see is that when we start to compare it, the amount of CCL2 does correlate to the amount of exposure they had. It also correlates to the amount of IBA1 positive cells and the amount of CD68 positive cells. And now for the interest of time, there's a lot more work in this study that I'm not going to be able to get into today. But the paper is on the bottom if you're interested in looking up how CCL2 is elevated. But what we can show is that CCL2 is elevated in the frontal cortex during CTE. It correlates with exposure to trauma, tau pathology, and it might be involved in bringing microglia to the pathology in lesion CTE. So maybe it's a therapeutic target. Maybe it's a potential therapeutic target. That might be this middle step here from exposure to RHI to neuroinflammation. Now, we already talked about a little bit what the inflammatory factors are involved here, and this is a very, very important field, and a very important part of my research is understanding these inflammatory factors. So now we're looking at, what is neuroinflammation? And that's a very complex question, but why do we really want to study it? And it might be very obvious, but I think it's important to state. We study these neuroinflammatory signals because we need to have a better understanding of the disease mechanism. And when we better understand the disease mechanisms, we can identify therapeutic targets. Because if you don't know what you're treating, how do you develop any sort of therapies? You need to have targets for that. You need to find novel biomarkers as well, because again, you can't treat CTE until you can recognize individuals that are alive that have CTE. So biomarker discovery is such a huge part of our work here. Now, neuroinflammation is a very, very complex topic. This is just a poster I grabbed off of one of the manufacturing sites of some of the processes in neuroinflammation. So whenever someone says neuroinflammation, they're referring to dozens, if not hundreds, of different processes. Pro-inflammatory, anti-inflammatory, blood-brain barrier activation, repair, regeneration. All of that's neuroinflammation. And before we really were working on neuroinflammation, we're looking at one or two different things. Like I mentioned, CD68 and IBA1. Those are very, very, very general markers, and we're not really able to understand the complexities of the inflammatory response when looking at one or two different things. It needs to be a very complex analysis that we take to understand this. And there are a couple ways we can do this, but what we chose to do was RNA sequencing. So RNA sequencing in these contexts of CTE lets us examine 20,000 targets at once. It lets us examine many facets of the immune response, but also many other responses in general. And it's one of the preferred ways to really understand the full spectrum of changes that are occurring in these degenerative diseases. And there are a couple different ways to do this. There's bulk sequencing, which is when we take whole chunks of tissue, so whole chunks of frontal cortex tissue, homogenize it, and sequence everything together. So that is an approach we've done several times in the past, and we've published several times on it, and it's a very good approach. We get a lot of really interesting data out of it, but there are some limitations that come with it. There's no cell type information. So you can't say a microglia makes something, a neuron makes something, an astrocyte makes something, because it's all ground together in a single analysis. So you're not going to be able to say that. Additionally, you lose a lot of subtlety in the fact that smaller changes might get drowned out by larger changes, and they're lost in the noise because you're looking at hundreds of thousands of things happening at once. Subtle changes might not be showing up, and that's something we're really interested in early disease. Maybe there's very subtle things we have to focus on that we can't see. So instead of doing bulk analysis, we do the individual cell analysis. And I'm sure several of you have read papers or have done single cell sequencing in the past, because it's definitely the hot issue now, or the hot experimental issue to be doing now. And we chose to do that. Using the single cell approach, just if you're not familiar with it, you take your whole chunk of tissue like you would for sequencing, but instead of grinding it all up, you process it to get single cells out of it. In our case, we use frozen tissue, and we got single nuclei out of it. Those single cells are then processed together to form essentially oil droplets with a single cell, and all the machinery needed for the reverse transcription and cDNA conversion within that one oil droplet. You do that for everything. And then all those individual droplets are barcoded. So then you can go back to say, if you're looking at a particular transcript, you see what barcode's on it, and you can pull that back to the individual cell from the individual case. It's a much, much, much more complex way to do this analysis and to get individual cell type data from these cases. So this is going to be work that we literally are just finishing now, or just getting, I should say, just getting the analysis going now. So I'm really excited to talk about it. And this is a project that's looking at early young stage CTE. So a lot of our work previously has been looking at the full range of disease, but what we're starting to get really focused on is what is the earliest possible event in the disease. Because I think that's where a lot of this matters the most, is this early event. You can look at someone who's 80 with 20 years of generative pathology, and the question is, how much can you restore? When you're thinking about more of the young side, maybe we can catch it early. We can restore a lot of cognition, prevent a lot of problems. So that's where this project is looking at. So we're looking at 3 different groups, 10 controls, 10 individuals with RHI with no disease, and 10 low stage CTE. And these individuals have very mild, if any, pathology. Well, they have some pathology if they're in CTE. All of these cases are under the age of 50, and the average age is 41. So again, very, very young, very, very mild disease. And this is where it's really important that we can look at this subtle distinction. We used the 10x platform to do single cell sequencing, and we isolated 10,000 cells per case. So if you're not familiar with single cell sequencing, this is sort of the first clustering you can expect to see from any sort of sequencing study. This is a UMAP, or some people use t-SNE plots to look at the overall clusters of their individual cell types. So every single dot on here is an individual cell. And now, because we've sequenced them all individually, we can look to see what they are based on particular marker genes. So we use marker genes to identify individual populations of cells. And we can see that we have a population of neurons here in the middle. We have endothelial cells, astrocytes, oligodendrocytes, and microglia. So all of our major cell types in the brain are represented. Now, there are a couple things we can do with these single cell studies. The first is a pseudo bulk analysis. So So just like for bulk sequencing, where you can examine everything at once, for some sort of pseudo bulk analysis, what we can do is just say, just give us the microglia and let's compare control RHI and CTE, just to look at general changes. So this is improvement on bulk sequencing in the fact that we're not looking at all these cells and trying to draw out a microglia signature. We're going to look at just microglia to look to see if there's any overall changes before we get into the nitty-gritty of the very particular data. And when we do the pseudo bulk sequencing analysis, we see that there are really, really interesting things that occur here. What's really important to point out is that it's not all everything is elevated, right? We don't expect that. So this is really showing us that neuroinflammation is not a pedal to the metal. Once you start having inflammation, it gets worse and worse over time. We see there are inflammatory elements that are up, and there are some that are down. There are some that are down we didn't expect to be down. So there are pro-inflammatory elements that are actually decreasing in early disease, and some anti-inflammatory elements that are increasing in early disease. So this is showing that inflammation is not a linear process. It's not even biphasic. It could be a multiphasic process where stuff goes up and down. And also what we're starting to see here is that individuals with RHI don't behave exactly similar to those that have CT. These are both compared to control. So there's a different response in different stages of disease. And that's something we want to better understand. So these are great plots to look at the overall genes that are changing here. But the best way to look at this as a cohesive data set is to look at gene ontology terms. So to look at overall function. When we do Go analysis or gene ontology, each individual one of these genes is assigned to a term, and we see what the predominance of those terms are. It gives us a better understanding of biology and lets us interpret these changes a little bit better. So first we looked at the Go terms of individuals who have RHI but no CTE. And the first thing that stands out as the highest, most expressed term is this eukaryotic translation elongation pathway. This is the metabolism pathway, which is really interesting that there might be a metabolomic change in individuals with exposure to trauma but no disease yet. But now we also see that there are several other immune changes occurring. So there's immune regulation that pops up several times, defense, adaptive, cytokine stimulus. So inflammation is occurring early in these individuals. And again, these are people that don't have disease yet. The only thing about these cases is they have exposure to head trauma. They played American football and potentially a couple of sports, I think, in this cohort, but they don't have any disease. And they have this elevation of metabolomic signal, and they have changes in inflammation. And these are young individuals too, mean age of 41. So young individuals played sports, have pathology ongoing, or have inflammatory responses ongoing. And then we look at individuals with CTE compared to control again, we see that there's a lot more inflammatory changes occurring. So it might be just at a higher level of inflammatory, but not the same level, just inflammatory change. The other important consideration is what is the difference between somebody with RHI to CTE? What does the transition look like? Is it just a continuum where someone with RHI just gets worse and worse and worse and they become CT, or there are different processes occurring there? And what it seems like there is, people with CT have more inflammation overall than individuals with RHI, but it's not that easy, and it can be pretty complex, it seems, too. The one really interesting thing that stood out to us is this TYROBP causal network in microglia that was elevated. So this is part of the DAP12 signaling pathway, which is involved in TREM2. Now, if you're any familiar with degenerative diseases at all, or microglia or any sort of immune category, TREM2 should be immediately evident. It's a very important signaling protein that's involved in glial phenotypes and inflammation, and might be involved in Alzheimer's. And we're seeing it being elevated here in the context of CTE. So it might be involved in the transition from RHI to CTE. Now, the most important thing that you can do, not most important, but one of the most interesting things you can do with the single cell sequencing data is the cell type analysis. So looking at different phenotypes, different clusters of microglia to identify particular changes that are occurring specific to disease population. So what we can do is take this microglia cluster out of here and recluster it to look at particular populations of microglia. So if you're unfamiliar with one of these UMAPs, generally the closer cells are together, the more related they are. And what we can also do, this is work by Nina Perez, who's done some phenomenal work with our single cell data so far, is you can say, well, show me all these different clusters that are most closely related to each other. So if it's a similar color here, it means it's in a similar group. And we've identified here it's 15 different clusters of microglia that exist. So these are different biologically relevant, potentially different clusters of cells that might correlate with disease severity. And when you actually plot them to look to see what phenotype they are or what pathologic group they are, you can end up seeing that red is control, RHI is blue, and green is CTE. There's this cluster of control cells down here. So this is where the majority of our control cells are. But then you have these outlying clusters of cells that exist only in individuals who have RHI and only individuals who have CTE. So right now we're identifying specific phenotypes of cells that might be specific to head trauma and involved in the translation to the disease cascade itself. And this is work that we're starting, from a couple weeks ago now, so it's still pretty fresh, and we're not going to have all the answers yet, but I'm happy to share some of the answers with you right now. Is that when you start to do this, you can look at particular sets of genes. Now, don't squint your eyes too hard at this. I'm going to zoom in just a second. But looking at these different populations, so these different marker sets here. So again, these different colors, we can see what genes make them express those different colors. What is the cause of that? What are the genes that are behind this? And for that, there's a group that we're finding, which is our homeostatic glia group. This is cells that express predominantly CX3, CR1, and NAV2, which are two important microglia homeostatic markers. And that is this cluster down here. So we're thinking clusters 5 and 6 are more of a homeostatic population of cells. Now we can also see that these other populations, so there's an individual population up here, which is a couple different individuals, is just RHI, and then we have another group of individuals that are just CTE. And when we look to see what genes are specific to them, what we can see is that in clusters 10 and 11, which is specific to just RHI, BIN1 is elevated. BIN1 is an AD risk gene, which is really cool to see in this microglia population. We're also seeing SPP1, which is an inflammatory gene. It's been published many times for inflammation, and hypoxia response. Again, these are people that don't have disease yet. These are people that have just played sports, and that's it. And they pass away and donated their brain, and they have AD risk genes elevated. And when we look at individuals who have CTE, we can see there are other reactive genes as well. So CD163 is involved in perivascular macrophages. Again, we showed previously that perivascular and peripheral macrophages might be involved here. We see there's lysosomal processing changes occurring, and there are other genes that are typically found with A beta we can see elevated as well. So this is all I have for this right now, but what we're trying to do is better understand the transition from this homeostatic cell type, how these populations of cells might become more to the CTE population. And we know CTE is very patchy. So something that's a very big caveat here is that we don't expect to see huge changes in this population, and we're not seeing huge changes here. The majority of our cells out of all cases exist down here in groups 5 and 6. We're looking at a very subtle change in microglia in young individuals with very limited pathology. So we don't expect to see such a huge change. And the fact that we can see changes occurring is really, really, really interesting, and it's very exciting. And we're still trying to understand what those subtle changes mean and apply that to the field as a whole. So overall, what the single cell sequencing data has shown is that there's multiple glial phenotypes related to these immune changes found in RHI and CTE, which is we've seen before, and that's just showing it more in depth in this single cell sequencing data. But not the same phenotypes in each group. So early RHI might be more metabolomic related or metabolomic related, and early CTE might be more phagocytic lysosomal. Now, we've seen this in bulk sequencing data. We published this several times already. There's work by Dr. Adam Labrador that was published very recently, where they could show that inflammation is different in early versus late disease. We see that with RHI and low stage disease. So inflammation is not a linear pathway, and I think that's very important to state, that starts low and ends high. It's a very cyclical, biphasic, multiphasic pathway with multiple cell types occurring. And we've seen the metabolism changes occurring too in other studies. I published a couple of years ago showing that individuals with RHI do have metabolism-related changes. So we're replicating our bulk data in our single cell data. And that's really exciting, and you always love to see when stuff replicates. So I'll just end here with future directions on where we want this research to go, and then I can leave time for questions if there are any. So what we have to do in the future is we really need to keep exploring these inflammatory factors that are involved in CTE. We have to dig more into the single cell data. which is a brand new data set that we're working through, and it's very exciting. We need to better understand what cell types are involved. Excuse me, we just worked in microglia right now, but if you remember that UMAP I showed before, we have every cell type that can be expressed in the brain pretty much captured. And we have to investigate what the neurons are doing, what the astrocytes are doing, what the oligos are doing, and they all might play a role in inflammation, and they probably do, but we have to investigate that. And then we want to also better understand CTE compared to other diseases, because CTE doesn't exist in a vacuum. Although many of our studies, we just use, quote unquote, pure cases of CTE, you can have CTE plus Alzheimer's, CTE plus Lewy body disease. And we have to better understand how these degenerative diseases that have their own cascades of effects work together. So before I end, it's incredibly important that I have to thank all our donors. We are incredibly grateful to them and their families for everything that they've done, and this work would not exist without them. So it's really important to point that out. And I have to acknowledge everybody in the BU Alzheimer's Disease and CTE Center, Dr. McKee, who heads our group, Dr. Torstein, Dr. Huber, Dr. Alvarez, Dr. Mez, Dr. Lasco. There's a lot of people that think this is a very small list. Morgan Butler and Nita Perez are spearheading a lot of the sequencing results, and Dr. Josh Campbell is overseeing some of that too. Sarah and Julia were very instrumental in getting that data as well, and they need to be congratulated for the amazing work that they've done with that so far, and of course my funding. And with that, I can take any questions if there are any.
Hi. Oh.
Sorry. Thank you, Dr.
Sorry.
Cherry, and I'll open it up for anyone to ask questions.
Oh, hi. I just had a quick question. Going back to what you were saying earlier.